gram staining, we want to distinguish between the gram-positive and gram-negative cells. So we will use the basic dyes, crystal violet and safranin. But you know, I think you should already know about the difference between gram-positive and gram-negative from the lecture which Dr. Young delivered to you. So the gram-positive cell wall, it has the thick peptidoglycan layer like this, and it can retain the crystal violet because of these thick peptidoglycan. But the gram-negative wall has a very thin peptidoglycan layer. So as you can see, it's very thin and it cannot retain the crystal violet because of these features, because of thin peptidoglycan layer. So how it works, the gram staining. We stain all of the cells with crystal violets, and then we will add iodine. Iodine form a complex with the crystal violet, and in this case, iodine is a mordant. Then we will rinse the cell with 90% of ethanol, which we call it decolorizing agent. The gram-positive cell wall hold onto the crystal violet because of the thick peptidoglycan, and these cells remain purple. But the gram-negative cell lose their outer membrane and the crystal violet because of the thin peptidoglycan, and it leaves it unstained, which means colorless at this point. Then, when we add the safranin, we then stain with safranin, the gram-negative cell wall will be stained pink. So, the gram-positive cell, when we add the crystal violet, it becomes purple. But the gram-negative cell wall, it's when we add the crystal violet, at this point, it becomes stainless and unstained. And then when we add the safranin, it will be stained to pink. So, if an old culture is used, old culture means more than 24 hours culture, the gram-positive bacteria lose the crystal violet during the decolorization step. And this means the culture will appear gram variable. But our culture is not more than 24 hours, so don't worry about it. And this can be caused by enzyme or autolysin, which break down the cell walls. So what is the autolysin? This is an enzyme degrading the component of the biological cells or a tissue in which it's produced. So today for gram staining, you need to write the lab report, so make sure you understand all the steps. If you have any question, let the TAs know. But you will be staining the E. coli, which is the gram-negative bacteria, and it has the rod shape, and also Staphylococcus epidermidis, which is the gram-positive, and it has the cocci shape. You already know that what is the rod shape and what is the cocci shape, I guess. So just one more time. The cocci shape is like this. The rod shape is like this. Okay? Then, we try to make, if you have a class, you will make 3 stains, one using the mixed culture and the other 2 with pure culture of each. But for today, we just make the mixed culture because you can see E. coli and Staphylococcus epidermidis in a one shot, okay? So we will view the stain mixed culture first, and then under the microscope. If the time permits, view stains of the pure cultures like each of them. But today we just going to do the mixed culture. And in your lab report, you just need to write the lab report from the mixed culture, okay? Because the procedure, everything's are same. At the end, you can see both bacteria, so don't worry about anything. You won't miss anything. Then, what is the procedure? First, you need to label slide with wax pencil, preferably, because if you label it with wax pencil, the ethanol could not rinse the wax pencil. Here is the slide. We will, for example, write it down as a mix. Okay? Then what should we do? Vortex the culture and smear a few loops of bacteria mixture. So here is the bacteria mixture, E. coli and Staphylococcus epidermidis. Okay? I will mix these two because we wanted to have the mixed culture. After I mix it, I need to vortex it. Okay, now we have the mixture. Oh, we have a lot, we don't need it. Anyway, then smear a few loops of bacteria on your slide and let it air dry. So first you need to sterilize your loop. Then after 30 seconds, you need to get a few loops of bacteria. Should be enough. And we need to let it air dry at least for 5 minutes. Now we let it air dry, and when it's dried, we need to heat fix the bacteria to the slides. Hold. What does it mean, the heat fixation? We need to hold over the incinerator for 1 minute, like this. We need to keep it for 1 minute. And after that, we need to stain the smear with crystal violet for 1 minute and then rinse it with water. Then, so here is the crystal violet. This is the crystal violet. We need to stain it with crystal violet for 1 minute. And then we need to rinse it with water. After we are doing that, we need to cover the smear with iodine and then rinse with water. So here is the iodine. We need to rinse it with iodine. So the concept of that is when we stain it with crystal violet, the gram-positive bacteria will keep the crystal violet and they become purple, but the gram-negative will be colorless. Then this is the reason why we add the iodine, in order to give the color to gram-negative bacteria. Okay, 1 minute is done. Then we need to add crystal violet. So here is the crystal violet. I'm gonna add it. Crystal violet. So I will put it here. It should be more easier. Okay. Now 1 minute is done for adding the crystal violets. We need to rinse it with water. Like this. After rinsing with water, we need to cover the smears with iodine. Here is the iodine. I will add the iodine. Should be like this. Okay. I covered the smear, and we need to wait for 1 minute. We will set the alarm for 1 minute. Timer. Okay. After we wash it with water, rinse it with water again, we need to cover the smear with 95% ethanol, which is the decolorizing agent step, and then we need to wait for 10 to 20 seconds and rinse it with water again. Then stain the smears with safranin. Here is the safranin for 1 minute, and then rinse it with water. So after we are doing that, the thing which is really important is after we wash it with everything, we don't want to we wanted to keep our smears, right? In order to do that, we need to blot dry with paper towel. 1 minute is done. Let me just rinse it with water, and then I will explain to you more. Here is water. Okay. Okay. After rinsing with water, we need to add the 95% ethanol. Okay. We will wait for this time for 20 seconds, and then rinse with water again. Ethanol. Okay. Again, water. Okay. This time we need to add safranin. Here is the safranin. Okay. We need to wait for 1 minute. Okay. So after we add the safranin and wait for 1 minute, we need to rinse it with water. But after that, we need to do the blot dry. Don't wipe, because we wanted to keep the smear. We don't want to just lose it. So we need to see the objectives under the microscope. Did you bring the microscope? Oh, okay. Here is the microscope, and I will take a picture of the end results. Make sure you need to label it and put it in your lab report, which the due date is 6A. And the picture that we are gonna send it to you guys is under 100x immersion oil. Make sure when you want to write the total magnification, it is under the 100x. And you can just put the picture and label it. And the other thing that you can do is you can draw it with your hands, write the and label the picture. There are two options for you. 1 minute is done. We are going to wash it with water. Okay. And then we need to blot dry. So I prefer to do it with the wipe because this is softer than the paper towel. What we are going to do is just like this. Okay. Put it under the microscope, and then I'm gonna send you the pictures of this, and also there is a lab report. By the end of the PowerPoint 5A, there is the guideline for the lab report. Here is the discussion, what questions you need to answer. Make sure to answer all questions, and for the last question, review supplement page 15, 16 and answer the question on the bottom of page about the lysosome. There is no question in the lab supplement. It was in the old version of the lab supplement. But your TAs will send you the question about this question and you need to answer it. Okay, so see you the other week.